Hello everyone, this section is for reproduction of bacteria. After learning this section, you should be able to describe binary fission as observed in bacteria and archaea, and compare the three reproductive strategies used by bacteria and contrast this with binary fission. Most bacterial and archaeal cells are haploid. Most of them reproduced by binary fission. Binary fission is a relatively simple type of cell division. See the picture. There is a young cell at early phase. The cell elongates as new material is synthesized, replicates its chromosome. We can see the cell wall, the cell membrane, and overall volume are all enlarged. DNA replication starts. Then the septum begins to grow inward as the chromosomes move toward opposite ends of the cell. Meantime, other cytoplasmic components are distributed to the two developing cells. Finally, the septum is synthesized completely through the cell center, creating two separate cell chambers, or we can call them progeny cells, each having its own chromosome and a complement of other cellular constituents. Some species separate completely as shown in the last picture, while others remain attached, forming chains, doublets, or other cellular arrangements. This is the process of binary fission. There are also other reproductive strategies. Several other reproductive strategies have been identified in bacteria. Some bacteria that have stalks, called hypha, reproduce by forming a bud at the end of their hypha. See the picture. It's a budding hypomonas cell. Certain cyanobacteria undergo multifission. The progeny cells, called biocytes, are held within the cell wall of the parent cell until they are released. Look at this picture. On the left is a drawing illustrating biocyte formation. On the right is a photo of biocytes produced by the cyanobacteria, the carpello. Some streptomyces form multinucleate filaments that eventually divide to form uninucleal spores. These spores are readily dispersed, much like the dispersal spores formed by filamentous fungi. Look at this picture. Spore formation by streptomyces. In color, the cells shown in the micrograph on the right are expressing a spore formation related protein fused to the fluorescent molecule M. cherry. Despite the diversity of bacterial reproductive strategies, they share certain features. In all cases, the genome of the cell must be replicated and segregated to form distinct nucleoles. At some point during reproduction, each nucleode and its surrounding cytoplasm becomes enclosed within its own plasma membrane. These processes are the major steps of the cell cycle. The cell cycle is a complete sequence of events extending from the formation of a new cell through the next division. The cell cycles of E. coli has been examined extensively. So let's see the cell cycle of E. coli. See the picture. Initiation of the cell cycle results from an increase in the mass of a cell, which also results in the accumulation of the DNA protein, which initiates DNA replication. As the cell readies for DNA replication, the origin of replication migrates to the center of the cell, and proteins that make up the replisome assemble. As DNA replication proceeds, newly synthesized chromosomes move towards poles, and the cell increases in length. When the cell reaches a certain length, initiation of septum formation is triggered and cytokinesis occurs, producing two daughter cells that have inherited one chromosome each. In this illustration, one round of DNA replication is completely before the cell divides in rapidly growing cultures. The generation times of about 20 minutes. Second and third rounds of DNA replication are initiated before division of the original cell is complete. Thus, the daughter cells inherit partially replicated DNA. So that's the cell cycle of E. coli. Okay, that's all for this section. Here's a word list for you.